Be talking about topic 3.3, cell division, which is about meiosis. A lot of this will be reviewed from 10th grade, but we're going to get into a bit more detail. So some topics to ponder as we go through this lecture is what's the purpose of mitosis, which we talked about in 1.6, versus what is the purpose of meiosis? So hopefully you'll be able to answer these easily by the end of the lecture. So let's review a little bit of the terminology that we learned from topic 3.1 and 3.2 about human chromosomes. Remember that all humans have diploid cells. So all of our cells, our somatic cells or body cells, are diploid. So they have a pair of 23 chromosomes, or we would total that to be 46. So we would call our cells, most of our cells, diploid. Now our sex cells are haploid, which would only be one set of each of the chromosomes. So when I say one set, that means if we were looking at chromosome pair 1 here, it would only be one of these chromosomes inside that cell. Remember, the formula for diploid is 2 times n. N represents the haploid number, so that would be 23 for humans, and then multiply that by 2, and that is 46. So we have 46 chromosomes, and they are all set up in homologous pairs. Remember, homologous means the same. So each of these chromosome pairs have the same genes on either chromosome, but they might have different versions of that gene. So a different allele, for example, like blue eyes versus brown eyes. So remember, a homologous chromosome is chromosome pairs that are of the same length and staining pattern and possess genes for the same characteristics at the same length. Some other terms we've talked about is the word chromatid. Now, a chromatid is one of 2 copies of DNA. So if we were to look at this chromosome here, one side would be a chromatid and the other side would be a chromatid. And if you were to divide it down the middle of the chromosomes, this would be a whole homologous chromosome. Now, a sister chromatid is when there are 2 identical copies of DNA. So this word pops up during mitosis and meiosis at different times, and it's because it's been duplicated, so it is now a sister chromatid. So if you look down here, this is a homologous pair of chromosomes. So here, remember, homologous means the same. This one came from the father, this one came from the mother, and this pair, for example, let's say it's pair 1. They contain the same genes for specific traits. Now, in meiosis, or mitosis, the cells need to replicate beforehand, right? So that it's double the same material. So if you look here at this homologous pair, these are exactly the same chromatids, so we would call them sister chromatids. And the whole goal is for it to have doubled in DNA, so when the cell divides, that each cell gets one of the copies. So we only use the word sister chromatids when the DNA has doubled right before it goes into mitosis or meiosis. The centromere is the pinched in location in the middle of the chromosome. The spindle fibers are what's going to attach to the centromere and pull it towards the centrioles, which is on either end of the cell. Okay, so the paradox. Human beings have 46 chromosomes in each cell. Your mom has 46 chromosomes in each cell, and your dad has 46 chromosomes in each cell. But you don't have 92 chromosomes in your cells. Why? Why is this? Why don't you have this? Why don't they just add together and make 92? Meiosis is the reason why. So if we look at understanding 1 here, meiosis does a process called reduction division, which is basically dividing in half from a diploid to a haploid cell. But it has to go through 2 stages in order to do that. So that's what makes it different than mitosis, which only goes through PMAT once, where meiosis goes through PMAT twice. So in this picture here, you can see this is the beginning after interphase in the cell cycle where all the DNA has doubled. So you're seeing a couple homologous pairs here. One from the mom, one from the dad. And they've doubled. So this would be a sister chromatids here, and this would be sister chromatids here. When they divide, it's going to divide into 2 diploid cells again, okay, but now they're homologous chromosomes. and then they've divided. Then in PMAT2 stage of meiosis, it's going to divide in half. And there's no interphase before this stage, so you're not going to have that doubling of DNA again. So the 46 won't become 92 before it divides. So the 46 will divide in half, and each of the sex cells will end up with 23 chromosomes in them. So one from each of the homologous pairs. So the product is 4 haploid cells. So that's something very different from mitosis, which is where you would start with 46 chromosomes in a parent cell, and it would double into 92 before mitosis, and then it would divide so that each of the body cells have the same exact chromosome information. Where meiosis is going to go through PMAT twice, and you're going to end up with 4 haploid cells. So we're going to go into this more in detail here, but I want you to just pay attention to the terminology. In meiosis 1, we talk about homologous chromosome splitting. So these are the homologous chromosomes splitting. In meiosis 2, we say a sister chromatid has split. So pay attention to those word changes, and that'll help you know which phase you're in when we talk about meiosis. Okay, so just to review the stages of meiosis 1 and 2, I've highlighted some important things you need to know about if you're asked what stage that this occurs in. So here's prophase 1 over here. The chromosomes condense in meiosis 1. And remember, it's very similar to mitosis in that way. The nuclear envelope's going to start to break down so that the DNA is more free. The chromosomes condense, and the spindle fibers are starting to form around the centrioles. A special thing, though, that happens during meiosis 1 is crossing over, which crossing over is when the homologous pairs start to share genetic information. And we'll go into that in more detail in a different slide. But this is part of what makes variation within human beings. Then it goes into metaphase 1, which is when the homologous chromosome pairs, so if you notice these are still in homologous pairs, but they have different chunks of coloring on them. That's because of the crossing over, which occurred from the different homologous pairs during prophase 1. And they're going to line up at this equatorial line of the cell. The way they line up is completely random, and this can allow for independent assortment, or the law of assortment, which means that any possible combination can end up in those 4 haploid cells because of how they randomly lined up. And we'll get into that more in another slide. Anaphase 1, which is right here, is when the spindle fibers attach to those centromeres and start to pull the homologous chromosomes apart. So one pair is going to go to this side, and the other pair is going to go to this side. The way that this is where segregations of chromosomes happen, and it's random which side either of them gets pulled to, which adds for more variation. And then telophase 1, which is where the chromosomes arrive at opposite poles of the cell, and the spindle fibers disappear. The nuclear envelope is going to start to reform before cytokinesis. So at the end of meiosis 1, or we could say PMAT 1, is the cell diploid or haploid? So hopefully you said diploid, because what's going to happen is we're going to end up with 2 cells with 46 chromosomes total in each cell. Because before meiosis 1, there was interphase where the chromosomes doubled, and it was 92. Okay, so meiosis 2. Meiosis 2 also goes through PMAT, or we could call it PMAT 2 for prophase, metaphase, anaphase, and telophase. And what's going to happen this time is there is no interphase or DNA replication happening before meiosis 2. So they're going to not do crossing over either during prophase 2. So the nuclear envelope starts to break down, and the spindle fibers and centrioles move into position. In metaphase 2, you're going to see that the chromosomes line up individually at the equatorial line, and then they're going to start to be in anaphase 2, be pulled apart. Notice I'm using the word sister chromatids at this point. So these are considered sister chromatids. And they're going to be pulled in half. So one part of the X will go to this side, and the other part of the X will go to this side, and you can see this one also is broken in half. Or one side of the pair will go to this side, and the other pair will go to this side. And then in telophase 2, the chromatids, because they're by themselves now, right? They're just one piece, so we call this a chromatid, is going to end up in its ending resulting cell or sex cell, which is haploid. So notice they only have half the amount of information in them. So it's going to be 23 chromosomes in each of these. And now these could either be eggs or sperm, depending on if the person is a male or a female. So the question at the bottom here says, is the cell a diploid or haploid cell after PMAT2? So what you're going to get is haploid cells. So just to kind of look briefly at the difference between meiosis versus mitosis, remember mitosis only goes through PMAT once. So here you're going to see before mitosis, there's going to be interphase where DNA replicates to 92 chromosomes. Those 92 chromosomes are going to be in pairs, like sister chromatids, like this. The sister chromatids are going to align along the equatorial line, and sometimes they call them daughter chromosomes. It's the same idea as sister chromosomes. They're going to separate. The chromatids are going to separate going either direction, and then you're going to end up with 46 chromosomes in each of these two cells, and they're going to be exactly the same as the parent cell was. So this is how we make new body cells when we need repair to our tissue, growth for a fetus as it's growing and making its own body cells in a mother's stomach. Meiosis is how we make our sex cells and how we get genetic variation. So here you can see at the beginning, the cells this is after interphases happened, and it's doubled to 92 chromosomes in homologous pairs. And what's going to happen is that there's going to be crossing over, so that's why they're twisted together like this, and pieces are being switched. So you can see here in metaphase, where they're lined up at the equatorial line, you can see different chunks on these homologous chromosome pairs. From the red is over here and blue is over here. And after they line up and crossing over occurred in prophase 1, they lined up randomly. They get pulled apart to the different cells. They go through PMAT2 now, and we would call them sister chromatids or daughter chromatids. And they're going to get divided once again, just as the same as mitosis at this point. And you're going to end up with 4 haploid daughter cells. So it goes through PMAT twice and only goes through PMAT once. Meiosis has variation occurring because of crossing over and how the chromosomes randomly line up and get segregated. And this creates genetic variation in our sex cells, making each one of these a possible different combination from another. So it's kind of like you don't know which one is, say these were eggs, you don't know which one a sperm is going to connect with, and that's going to create even more variation because all of these had different possibilities, and you don't know which one you're going to end up with. So if you look at them, like this blue chromosome has a little red, this red chromosome has a little blue, they're all slightly different, and it's a chance which one you're going to get. Okay, so just to kind of rephrase, what's produced at the end of meiosis? You're going to have 4 haploid daughter cells. The having of the chromosome numbers allows sexual life cycle and fusion of gametes. So it's important that our sex cells are only 23 because when they combine with the other sex cell from your partner, it's going to create a new combination. So sexual reproduction allows for tons of different combinations of variations because each of these sperm have their own unique combination, and whichever egg is released each month from the female has its own unique combination. All of her eggs are going to be slightly different, and all of these sperm are potentially different. So when they combine, it's going to create 46 chromosomes, 23 pairs. Each homologous pair will have one from the mom and one from the dad. And then once it'll start to develop and do mitosis and form into an embryo or to a fetus over time. And then this embryo will start doing meiosis and make its own sex cells for later in life. Just to come back to this, remember, women usually have created all their eggs, so they're done going through meiosis by the time they hit puberty. And men go through meiosis all the time. As long as their body can create sperm, their bodies are constantly making sex cells, where women stop doing that by puberty. So this embryo will start to go through meiosis and make its own sex cells for later in life. It starts around that time. Okay, so what happens during interphase before meiosis begins? It's important to remember that before mitosis or meiosis, there is the full cell cycle. So the G1 phase, the synthesis phase, the S phase, the G2 phase for more growth, and then mitosis or meiosis. So in interphase, you're going to get DNA replication. This is when the DNA is doubled to make sure that we end up with the right amount of chromosomes. So always in interphase before meiosis 1, or before mitosis, it's going to double the DNA through DNA replication, which is what this picture is showing. And it's going to be 92 chromosomes. So that after the division of the first meiosis, there'll be 46 in each of the two. And at the end of mitosis, there'd be 46 in each of the two. So something that makes meiosis unique from mitosis, remember, is crossing over, and this occurs in prophase 1 of meiosis 1, not mitosis. So what happens is non-sister chromatids from each homologous pair intertwine, and there's a reciprocal exchange of genetic information. So when you look here, this would be like a homologous pair from the father. This could be a homologous pair from the mother. And they start to intertwine with each other, and they literally tear pieces off and kind of accept each other's genetic material. So here you're going to see that the pieces have switched. So some of the green is on this homologous pair now, and some of the yellow is on this homologous pair. So this is creating a unique combination between your parents' cells that were genetically given to you. And this means that you're going to pass on a new genetic combination to your offspring, because this is happening in your body when you start doing meiosis. So your parents' chromosomes then go through crossing over and create a new combination, which is you, which is your cells. And when your body starts doing meiosis, you're going to break down the chromosomes through crossing over and have a new combination with whoever your partner is. So crossing over is what contributes genetic variation in humans in sexual reproduction. It's one of the ways. Also, the law of independent assortment is what allows for genetic variation. So if you see how the homologous pairs line up is different in each of these pictures. So you could have this blue pair and red pair line up this way, which is the same here, but then you see the other chromosomes, homologous chromosomes, are different. They're opposite. And this means which ones end up on either side when they divide are going to be unique. So you can see it created this combination here later on within the cell division in metaphase 2. And then you're going to see there's a combination for each of these that is unique because of that. So each of these sex cells have a different combination that could potentially be the one that, if these were all eggs, that a sperm picks. So even just the way the chromosomes line up in the middle during metaphase 1 and metaphase 2 is called the law of independent assortment. So they're independently sorting themselves out. And this is random, and that allows for genetic variation. So how many chromosomes does each daughter cell have at the end of meiosis 1? So hopefully what you're remembering is at the end of meiosis 1, there are 2 daughter cells and they each have 46 chromosomes in them. So we're not done yet because the haploid cells, there's only 23. And that's at the end of meiosis 2. Okay, so it's important to recall where the genetic variation is coming from in the process of meiosis. So through crossing over in prophase 1, through random orientation, which is happening in metaphase as well as anaphase as they get pulled apart. So we call that random orientation. And at the end, you're going to produce 4 haploid cells, and they're each going to have their own new combination of alleles. They're going to each be different from each other, and that's genetic variation or variety. And that's what's special about sexual reproduction, is it allows for this variation of traits, and those traits might help a species or an organism live better or be immune to something that someone else is not later in life when environments change or new sicknesses arrive. Okay, one thing that can happen during meiosis is called non-disjunction. Now, when we looked at karyograms before, or karyographs, this is what they're looking for. They're looking for any time that an extra chromosome pulled away and went into another one of the cells singly or as a pair of three. So in this picture you can see here, everything's going fine in the beginning. Okay, so this is prophase 1. And they start to be pulled apart here in anaphase, and you can see in anaphase 1, what's happening is that this pair, both these homologous chromosome pairs are getting pulled to this side. So that means after anaphase 2, both these potential eggs are only going to have one chromosome in them, and these potential eggs are each going to have 3. So these would be examples of how someone can end up with an extra 21st chromosome, and we notice a pattern. There's about 5 different chromosomes that this happens to sometimes. So thus an egg or a sperm can contain an extra copy of chromosome 21, and a fertilized egg can have 3 copies of chromosome 21. So this occurs in meiosis 1 or 2. This can happen. So this would be an accident in the process, or a mistake in an error in the process, and it can create some detrimental effects in the end. This would be an example, Turner syndrome, of non-disjunction when someone inherits only one an egg cell with only one chromosome in it. So in this case, they only have one X chromosome and they don't have the Y chromosome or another X chromosome. So this features short stature and infertility. So you can see here this male has a gamete with 23 chromosomes, and then this partner, this gamete, has 22 chromosomes. So you're going to end up with monosomy, right? When their egg and sperm come together, and that's going to leave this cell with one missing chromosome. For its 46, it'll be 45. And so you can see the label there, 45X. So some methods for karyotyping. We never talked about this in our last chapter. We talked about how to read a karyogram, or how to do a karyotype and to read a karyogram. So the ways that they get the DNA for, usually they do this to look at a fetus and to know not just their sex, but you don't need to do this to find the sex, but to look for genetic diseases. So they would insert, there's 2 ways. So the first one is amniocentesis. Now amniocentesis is where they insert a needle into the uterus. So here in the picture you can see that happening. And they use ultrasound to try to make sure they can see where the baby is so they don't hit the baby. And they take the amniotic fluid, about 10 milliliters, and extract it. And this is the liquid that bathes the fetus is living in. They run biochemical tests of the fluid to identify certain chromosomal defects. So some tests, though, can require that cells be cultured, which can take several weeks. So they're growing the cells, basically, before they can be tested. This can be performed around the 14th and 16th week of the pregnancy. And some risks of doing this test is that you can have a miscarriage, and which means the baby will not develop properly and will stop basically growing. And usually the woman will bleed out and release the baby like she has a period. And this can happen to every 1 in 200 to every 1 in 400 births by doing this test. So some people choose to not do these tests to check for genetic diseases, and they hope for the best, right? So chronic villus sampling, or CVS, is the other type of way to do a karyotype. So the chronic villi are right here in this image. And they're like microscopic finger-like projections which surround the developing baby, and they eventually are going to form the placenta. So they insert a narrow tube through the cervix. So here is the narrow tube here. This is their opening up the cervix, and then they insert this narrow tube. And they basically take out a little fetal tissue. And the cells of the chronic villi are rapidly multiplying, so that means the test can give you results in 24 hours. It can be performed as early as 8th to 10th week of pregnancy. So this is early enough if there's any issues that someone can make a decision about termination of pregnancy. If, for example, the baby won't come to full term because of the genetic disease they have. And the miscarriage rate, though, because this could actually cause a miscarriage for someone, and it happens every 1 in 100th to every 1 or 200th birth. So it's a higher risk. So some people choose not to do this test as well. And you might ask, why would someone want to know? It is the idea of wanting to know if their baby will have a genetic disease and what they need to do to cope with that, or if the baby will even come to full term. And then they do have that option of having an abortion if they believe in that and if they would choose to do so. But it is a choice, and a lot of people choose not to do these two tests. All right, so that is it for the meiosis lecture. What I want you to do to kind of review the topic is to complete the M&M table, which is in the link on ManageBac. So you're going to complete this, and you're going to just kind of compare mitosis versus meiosis. And then I want you to read an article about risks of older pregnancy. This link is also in the assignment for this lecture on ManageBac. And it kind of talks about, why do we talk about when women are in their 40s or hit 40 that they shouldn't have, and even with males, that they shouldn't have become pregnant anymore. Or not that they shouldn't, but that they're putting themselves and maybe the baby potentially at higher risk. So take a look at this, and we'll discuss this in our next class. Let me know if you have any questions, and please put your notes in ManageBac from this lecture when you're done. Have a great day.